introduce our next presenter. Presenting next is Q Therapeutics, and presenting for them is Deborah Eppstein, their CEO. They're located in Salt Lake City. Deborah?
Well, thank you for sticking around for this presentation. What I'm going to talk to you about today is quite different from what you've heard in the other presentations. I'll start off by saying we're now a public reporting company and expect to have stock trading by the end of the year. So, of course, I am showing you our safe harbor statement. So why should you be interested in Q Therapeutics? We're doing something that we really do believe has the ability to revolutionize how central nervous system diseases are treated. What we're doing is using methods that have developed over tens of thousands of years by the human body that are safe and effective in maintaining normal function of the central nervous system. So they're natural cells in their native environment. This reduces off-target effects and doesn't require engineering. The cell therapy field in CNS really is an emerging sector, and very importantly, we have strong patent protection. Interesting how they slowly fade in here. So our mission is to improve the lives of the millions who are suffering from these debilitating neurodegenerative diseases and injuries for which there are no effective treatments. Our initial targets are focused in the spinal cord, looking at ALS, transverse myelitis as a stepping stone to multiple sclerosis and spinal cord injury. But we would certainly like to look at a variety of other diseases once we have appropriate partners and funding. Okay, so what are we doing? We're developing our product, which we've appropriately called Q cells, which are glial progenitor cells. What do glial progenitor cells do? Well, if we look at, CNS 101 is what I call it, we have two basic types of neural cells. The neurons, which are, the cells that transmit the signals, and then the other cells, the glial cells, which actually are the majority of the cells in the CNS. And the glial cells are what enable the neurons to function. They produce two general types of cells, the astrocytes that have many important functions. Some of them include producing growth factors, not just one growth factors, but a whole range of growth factors, removing toxins, and then the oligodendrocytes, which are the cells that wrap their processes around the neuronal axons to produce the insulating myelin. I think it's an important point that a lot of people don't realize is that the myelin is part of a living cell. So what we're looking at is restoring the function and improving the function of existing neurons, not trying to replace the neurons with all their billions of connections, which we view as a very daunting task. But we think this is a much more achievable way to benefit many neurodegenerative diseases. Okay, this slide, a cartoon, a little busy, but the main point of this slide is to show how our cells relate to other cells in the central nervous system. So the neural stem cell shown, there's a pointer here, right, can produce both neurons as well as the glia cells. What we're doing is starting with the cell that is only a glial progenitor and only can produce the two glial cell types. And this is a distinguishing factor from us and other companies that are working in this area who are working with neural stem cells. We have 15 issued patents, many more pending, so very strong protection to 2030. In vivo, we can show that if we implant our cells, we produce only the two cell types that I mentioned, the astrocytes, which are shown here stained in red, and then down here with the green are the oligodendrocytes, and that is myelin production. This is in the shiver mouse model, which does not have endogenous production of myelin by myelin basic protein. So we've looked extensively to see if neurons are produced by ourselves, and as predicted, we do not get neuron formation. Some data, looking at, again in the shiver mouse model, is that we can get long-term protection of animals in a disease where the entire CNS has defective myelination. This was published by Steve Goldman a few years ago, but he has shown that some animals survived out to a year. Control animals are dead after a few months. But what's shown here in red is staining for the human cells, and this is showing that the human cells widely migrate throughout the brain, and shown here with the green is staining for myelin produced by the human cells. We have replicated this work and have shown that we can get over 80% long-term survival of these animals. So this really shows that not only do we get myelin production, but it is functional. Looking at the time course, which is, I think, quite important, we can see that after 2 weeks, we have a very small number of cells, shown here again by the green as the human cells. The red is staining for the astrocyte marker, and down here, red is staining for myelin. So no myelin at 2 weeks, small amount of cells, near the injection site. By 6 weeks, they've migrated out. We now get more astrocyte production as well. We can see the beginnings of myelin production. And now by 13 weeks, we can see that there is extensive both migration as well as expansion. We get about a 10-fold expansion, and again, very importantly, extensive myelin production. So these cells follow the endogenous cues in the CNS to do really just 3 things. They migrate within the CNS, they expand, and then they terminally differentiate. This allows us to need fewer cells because of the expansion, but also fewer injection sites because of the migration. We've looked at traumatic spinal cord injury in collaboration with Itzhak Fisher, and have shown that when we treat it as late as 9 days after the injury, which is later than a lot of labs look at, we saw benefits both for cyst formation as well as glial scar formation. These are very important because both of these prevent cells from getting in there and healing the injury. And we also saw benefits on bladder function. With our collaborators at Johns Hopkins, this is Nicholas Maragakis' laboratory. We've looked at ALS, animal model using the rat SOD1 model, and using, in this case, rat glioprogenitors. We had treated the animals after disease onset, and we see here on the left panel almost a 2-fold increase in time to survival after disease onset. These cells were injected in the cervical area. That was chosen because it is loss of respiratory function, which really kills the animals as well as humans. And Dr. Maragakis was able to show that he actually preserved function of the phrenic nerve. And on the right, he looked at motor score of the forelimb. And again, those motor neurons are in the cervical area. So that's very compelling data. And in fact, this will be our first clinical target. We're looking at treating ALS patients. It's a huge unmet medical need. The incidence of ALS is almost 6,000 a year, comparable to the incidence of MS. It's just patients don't live very long, so the prevalence is much lower. It counts for us, oops, pushed the wrong button there. Can I go back? It accounts for 1 in 400 deaths in the US. I find that a very striking number, and in the military, Gulf War veterans have twice the incidence of ALS. It's known that in ALS, the motor neuron death is what is causing the symptoms, but the death of the motor neurons is accelerated by non-neuronal cells, and particularly diseased astrocytes. So our goal is to replace diseased astrocytes with normal, healthy astrocytes via our glioprogenitors, and that's what we showed in the animal models. So Q operates in a semi-virtual mode. We work with various academic as well as institutions around the country. For the manufacture of our product, we're using University of Utah has a GMP cell therapy facility. We've produced the antibody that we use for purifying these cells under GMP conditions with GBI. Preclinical studies, we've worked again with Johns Hopkins. We're working with Emory University on the device, and MPI Research for our animal safety studies. And then clinical trials, we'll be working with both Johns Hopkins and Emory. We have a stellar team of scientific advisors and collaborators. Many of you know Mahendra Rao, who was our scientific co-founder back when he was at University of Utah. He is now the director for the NIH Center for Regenerative Medicine. Doug Kerr, Itzhak Fisher, Nicholas Maragakis, Ian Duncan are some of the others that we are actively working with. So to summarize then, we're developing a novel cell therapy product, which is a natural product we believe is the best in class in this area to treat these debilitating diseases with no current treatments. Preclinical studies have shown very promising results in multiple diseases of the CNS. We do have strong patent coverage to 2030, strong leadership team, and strong group of scientific collaborators. We have been validated both by industry and the scientific community, both by a $4 million NIH grant, so that's very nice non-dilutive funding, as well as an investment by Cephalon, which is now Teva. The regulatory pathway we have discussed very extensively with the FDA and are looking to file our IND next year. As we talked about, this huge market opportunity, and lastly, we are now a public company, which with trading to commence hopefully by the end of the year. So that is it. I think I stayed within the time.